Okay, well, thank you everyone for joining. My name is Ellie Hayes. I'm the Executive Director for Missouri Cures Education Foundation, and we're so excited to bring you our Lunch with a Researcher event. This was an event we held quarterly for many years, and I'm really excited to bring it back for 2025. So thank you all for being here, and thank you, Dr. Nichols, for joining us today. I'd like to introduce to you Dr. Michael Nichols, who is a professor of chemistry and the director of Biochemistry and Biotechnology Program at the University of Missouri St. Louis. He's going to be talking today to us about his research in Alzheimer's. So without further ado, I will turn it over to you, Dr. Nichols.
Thank you very much, Ellie. I really appreciate the invitation. I'm always happy to talk about uh, the basic biochemistry of Alzheimer's disease and the research that we're doing. Bear with me for a second while I share the screen here. I do have a PowerPoint, and I think it's really important to see some of these images, and hopefully that will help with everyone's kind of understanding of the pathology and etiology of Alzheimer's disease. So what I've got planned, and welcome to everyone who's here. It's great to have you. What I've got planned is a fairly rigorous background of Alzheimer's disease. And what I want to do is kind of bring everyone up to speed on what's happening as Alzheimer's disease is triggered and as it progresses. And that'll kind of lead into some of the current diagnostics and therapeutics that we have going. And then we'll all kind of be on the same page. I've got a little bit of our research. I'm not going to overload everyone with that, but hopefully it'll spark some questions, and I look forward to answering any of those. So I think the people here today probably understand the problem with Alzheimer's disease. It's the most common form of dementia. It attacks the brain, and we end up losing neurons to the point where the disease is fatal. And there's a couple areas of the brain that it attacks, and one of those is primarily the hippocampus. Let me see if I can get my laser pointer going here. Here we go. Hopefully you can see that. And the hippocampus is very important for our memory. And in particular, short-term memory is impacted the most. But it does spread to other areas of the brain, and ultimately you lose neurons, and so the brain shrinks pretty dramatically in Alzheimer's disease. Okay, very disruptive. I think most of us probably have encountered Alzheimer's disease in our own families or with some of our friends. Okay. There's not many other diseases that are, I mean, they're all horrible, but that impact us as closely as Alzheimer's disease does. So these are some statistics for AD. I think, I'm not trying to scare anyone, but I think we all know that the number of cases has been rising. Part of that is due to better diagnostics, and part of that is due to the aging population. Okay, but the real thing I want to point out is how much of it impacts every one of us. And it's not just older people, but it's the entire spectrum of ages, because even the young people are having to deal with the consequences of the increase in AD in the population. One that always jumps out at me is if you have AD at age 80, 75% of age 80 is in a nursing home. For non-AD, it's only 4%. So it's just a dramatic difference in the type of care that's required. And here's one of the most striking things. We've made a lot of progress in handling some of the other diseases, and all of these are important, but we've actually had an impact on these. AD and the other neurodegenerative diseases, not so much. And so when I say other neurodegenerative diseases, I'm talking about things like Parkinson's and ALS, Huntington's diseases, because at the root of those involves protein aggregation. And I'll spend a little bit of time on that in the next few slides. So I really view this as the challenge for this particular century in tackling AD and these other protein aggregation diseases. So AD is considered a plaques and tangles disease. And it's these plaques and tangles that I'm going to point to in the next slide so that we kind of understand what's going on in Alzheimer's disease and the timeline. Okay, so the plaques in this kind of depiction here are these fluffy things, and then the tangles are inside the neurons. But we'll look at this in a little bit more detail. Okay. So let's focus on the plaques right now. So maybe we've heard the term senile plaques. They accumulate in the brains of patients with Alzheimer's disease, and they're primarily composed of a protein called the amyloid beta protein. Now, A beta for short, and that's what I'll call it from here on out, forms these plaques, and it's outside of the neurons, okay, and it's in the brain. So this is an actual slice from an Alzheimer's disease patient. If we look at these plaques in great detail by an electron microscope, we can see that all of the A beta is now in these fiber-like structures in the plaques. Okay, and so it's not just the A beta accumulating, it's accumulating in this fiber-like structure. So what that is called is an aggregate, so an aggregation of the amyloid beta, and then it accumulates in the brain. And that's important because we all produce A beta every day, and it gets cleared every day. And I'll talk a little bit more about that. But it's only when it begins to aggregate into these fiber-like structures that the disease starts to occur. So these are in AD patients. These are in mice that have been engineered to get Alzheimer's disease. And what we can see in the A beta plaques are in blue, that all of the neurons in green that are in contact around those plaques are damaged. And we call those dystrophic neurites. But essentially, they're neurons that have been degraded and damaged around the plaques. To add another level of complexity, around the plaques in the brain are the immune cells of the brain called microglia. And in this image, in green, are all the microglia, and they cluster around the plaques, trying to get rid of the plaques. They're very inflammatory, and so what that creates is a chronic inflammatory environment in the brain. And as most of us know, AD is a long-term progression. So this goes on for quite a while with this chronic inflammatory environment. So that's all about the A beta plaques. Where does this A beta come from? And here's where it gets really puzzling, because even at this stage, we still don't know a lot of the normal functions of these. So A beta comes from this much larger protein here called the amyloid precursor protein. We have APP in our brains and in other tissues of the body, and we don't fully know the function of this APP protein. A beta is in green, and it actually gets produced by two enzymes that cleave this fragment out of the protein. We have these proteins in our brain and in other areas of the body. We don't really know the normal function of those proteins, these enzymes that cleave the fragment. So here every day we produce this fragment. It circulates in our system. It gets cleared every day, and yet we still don't really know a lot of the normal functions and why we produce this protein. Now, there's a lot on this slide, and what I'm highlighting are a lot of mutations that are fairly rare, but around the world, that cause early onset Alzheimer's disease. And so this would be AD before probably the age of 50. And many families around the world know that they have these mutations, and they know in their family that they get early onset AD. So these mutations come into play because these families have been incredibly gracious in sharing information with the researchers and even tissues after death that allow the research to go on and try to understand how these mutations impact AD. Because for most of us, it's the late onset or the sporadic AD that affects much larger part of the population that we're interested in. We want to know the genetics. We want to know what kind of environmental factors might be involved in AD. Okay. All of these mutations either increase the level of A beta that's produced or increase the amount of A beta 42. So these little letters represent amino acids. So if you're a biochemist, you know very important properties of these amino acids. But just the addition of these two amino acids for A beta 42 really implicates that form of A beta in Alzheimer's disease. It aggregates much more quickly, forms these plaques much more rapidly. All right, a lot of questions there. What about the tangles? So there's another protein called tau and we know the function of tau inside the neuron. Okay. Tau is a microtubule stabilizing protein that really helps the dynamics of neurons and all the processes that are involved in the neurons. So when the tau tangles start forming, not only do you lose the function of that protein, but it also forms these filament-like structures inside the neuron. And that becomes quite toxic to the neurons. So this is kind of an example of a normal tau. These are the axons of neurons. And so neurons have these long processes that communicate information from the end of the neuron back to the cell body. And tau is very important in that communication process. When the tau filaments start forming, it damages these axons. So you have whole losses of the axons, and the tau is in a kind of a unique aggregated state. And so it doesn't do what it should normally be doing. In addition, these filaments, again, are toxic. Okay. So you have the A beta and the tau. Both are in an aggregated form. So what's the timeline between those two? Well, the first thing that happens is the A beta begins to accumulate. Okay, that's in red. What follows right along with that is the activation of these immune cells in the brain, microglia, astrocytes. So the inflammation goes right along with that. At some point later on, the tau tangles start forming. Okay, so it doesn't happen at the same time. Once the A beta is accumulated and the tau tangles are forming, then the neurons really start to be susceptible to all of those insults that are going on in the brain. Down here on the bottom is the cognitive impairment, so dementia. And what you see is that you don't really start seeing symptoms until a lot of this has already happened here. And the symptoms correlate pretty closely with the tau tangles. But you don't get the tau tangles without the A beta accumulation. So what does that mean? It means that A beta is the trigger for the disease, but tau, when the tangles start forming, really hastens the progression of the disease and the symptoms. So that leaves us with a ton of questions. And all of these are being actively researched right now. So what's going on before all of this happens? Okay. What happens between the A beta accumulation and the tau tangle formation? And then what are all the mechanisms of toxicity? How are these two proteins damaging the neurons? A lot of questions, but a lot of areas of research here. So I should spend a little bit of time on the aggregation of A beta. And we can talk about tau, but for today, since my work is mainly related to A beta, I'll just spend a few minutes on this. Much of this has been done in the laboratory. It's very difficult to follow the aggregation process in living humans. Okay. But typically we all make the A beta, and it's called what's called a monomer. So it hasn't begun to aggregate. But at some point, it begins to aggregate into these oligomers called protofibrils, and then ultimately the fibrils that are found in the plaques in the brain. There's lots of things that impact this process. It's not a clean process. There's a lot of things that happen, but these structures can be observed in the brain, okay, and ultimately the plaques that form. Okay. We have ways of studying this in the lab. This compound down here, thioflavin T, becomes very important in the next slide when I talk about diagnostics. But we can study this aggregation process, and we've done that for many years in my lab. And it kind of forms this S-shaped curve as it goes through this process. So what do we know? We know a lot about the structures. And for the protein biochemists out there, this beta sheet structure that's an aspect of proteins when they fold. But all of these aggregated structures are highly enriched in this kind of beta sheet structure. The fibrils here are insoluble, so they deposit, but all of these precursors are not. They're quite soluble, and they can diffuse around in the brain. So these fibers and fibrils that are formed in the lab, very similar to the ones that you see in the brain. And that kind of filament or fiber-like structure looks like this. And this is actually, this model here is derived from a very sophisticated type of scientific analysis that can look at structures of things. Okay. All right, so what about diagnostics and therapeutics? Well, on the diagnostic stage, we now have the ability to image amyloid beta accumulation in the brain. Okay. And so this is a compound called Pittsburgh B that's based on that thioflavin T structure that we use in the laboratory all the time. And so it's carbon 11 labeled, so we're able to do a PET scan on people that have been diagnosed with AD and want to look at the accumulation. And so what you see in the AD brain is a lot of amyloid or A beta accumulation in the brain compared to a normal control brain. So that is it's kind of course, it's a PET scan. You have to be administered this PIB compound. The radioactivity goes away pretty quickly, so you have to have the PET scan in about 20 to 30 minutes. And it's invasive, so it's not what everyone's going to do on an annual checkup. There's also a very good cerebrospinal test, again, pretty invasive because it involves a lumbar puncture, but it has a pretty high diagnostic accuracy for AD. And it involves the tau protein that I was talking about on the previous slides. So tau gets phosphorylated, and what that means is a phosphorus group gets added to the tau filaments. Okay. So you can measure that, and the total amount of tau that you have can be measured, and the amount of A beta 42 that you have. So this is actually a pretty accurate cerebrospinal test that can differentiate AD from other dementias, and that's important because there's lots of other things that cause dementia that can be mitigated. And so it's very important to distinguish whether you have AD dementia or something else, like maybe you're mixing medicines or something that can be pretty quickly changed, or maybe it's some sort of nutritional thing that can be changed. So you really want to be able to distinguish that from AD. So what would be something that we're all striving for, and what kind of progress has been made just in the last year or two? And so this is a blood test here that looks very promising. And that also is looking at a phosphorylated tau and the ratio between A beta 42 and 40 in the blood. So this would just be a blood test. So it's very preliminary now, but promising. And there's a large study going on. And so these percentages are not just based on a few people, but the diagnosis accuracy is very good, 91%, even better than if these folks went to a primary care or a specialist to be diagnosed with AD. So very promising, still some work to do, but hopefully that'll be out in the next few years. And that I hearken back to that timeline slide that I showed earlier. So the earlier diagnostics that we can get, and so this is not very early, but if we can push that earlier and earlier with an easy test, that is also going to enhance the possibility for therapeutics. So what about the therapeutics? So right now, we've got quite a few medicines that will not affect the progression of the disease, but they'll treat some of the symptoms. And these are very important. But what we really want to get after are things that modify the disease. So right now, there's a couple of antibodies that have been approved over the last 2 or 3 years that actually impact the disease progression. They don't stop it at this point, but they slow the cognitive decline. Okay. Both of these are targeting aggregated A beta. Okay. And they're selectively targeting the fibrils and protofibrils that I talked about before. Okay. So these are being administered right now to AD patients. Happy to talk about these in just a little bit. So what about the work that we do in our lab? Okay. Well, we study the biophysics of A beta aggregation. Some of the things that you've seen on the previous slides we've contributed to in different aspects. We're also interested in how the A beta aggregation process impacts neuroinflammation. And upon doing that, we're interested in different inflammatory pathways that are upregulated in Alzheimer's disease. So we do that in the lab by focusing on immune cells, whether it be microglia, monocytes, macrophages. These are all cells that we have in our body. And we use those as cell model systems to study a lot of different things and how A beta interacts with these cells and activates them to cause an inflammatory environment. Okay. So what do these experiments look like? Well, we followed A beta aggregation over time, and then we looked at when it could produce the production of an inflammatory molecule called TNF alpha. TNF alpha is highly implicated in things like rheumatoid arthritis, but it's involved in all inflammatory processes in the body and also in the brain. So we measured this TNF alpha, and we saw that at an intermediate time point, it sparked the most production of this molecule. And we used a technique called atomic force microscopy, which allows us to look at these species, was very reminiscent of these protofibrils that we saw that I presented earlier in the slides. So we can prepare and purify these protofibrils in the lab, and we can do this at physiological conditions. We can characterize these, so I wouldn't expect anyone to know what circular dichroism is, but this red line is indicative of that beta sheet protein structure that I was talking about. So the protofibrils have that, but the monomers don't. The monomers are not toxic or not pro-inflammatory at all, but it's only when they adopt this type of structure. And we can use electron microscopy, another high-resolution technique, to look at these much shorter diffusible protofibrils and the much longer insoluble fibrils. So these are the scale bar, 100 nanometers, that's very small. But I think you can appreciate the protofibrils are much, much smaller than these long fibrils that cover the whole length of the image. And when we compare these two in a variety of studies, we see that the protofibrils will bind to the immune cells much, much better. So this is a confocal microscope image, and we can use different fluorescent labels to follow the species that we're trying to study. And so the fibrils didn't really bind, the monomers didn't really bind. The blue is the nucleus of these microglial immune cells. And you can see that the protofibrils bound to the outside of the cells. Not only that, but they induced a much higher level of this TNF alpha pro-inflammatory molecule again, compared to the monomers and the fibrils. Furthermore, they get inside the microglia much, much faster than the other A beta species. So we can see that after 10 minutes, the microglial cells have already taken up the protofibrils. And that's what these cells are supposed to do. They survey the brain. They try to get rid of things that shouldn't be there. Okay. So I'll go through the next few slides really quickly because I want to leave lots of time for questions and answers. But because these protofibrils have done so much in so many of our studies, we actually developed an antibody that was selective for these protofibrils. Okay. So we immunized a rabbit with the protofibrils and looked at the serum and found that the serum had generated antibodies that were selective for the protofibrils over the monomers and the fibrils. And this is all A beta protein. Okay. So we can do different studies to again show that selectivity for the protofibrils over the monomers and the fibrils. The fact that this curve is shifted to the left of the other two means that it has a much higher affinity for the protofibrils. And we can look in transgenic mice that have been engineered to get human Alzheimer's, and we can see that the antibody that we have recognizes different things from really conventional antibodies of A beta. So in this case, it was totally different. And in other cases, there was some overlap between the green and the red of the conventional antibody. And you would see that in yellow. Okay. In other cases, they were totally different. And what we also see in the mice that have not been engineered to get Alzheimer's disease, they don't have these plaques that show up in the mice that have been engineered to get Alzheimer's, and our antibody recognizes these quite well. So if we blow that up, our antibody would be in the green, the conventional antibodies in the red. And you can see that there's whole different elements in the brain that our antibodies recognizing. So we've developed a monoclonal antibody, very similar to the serum antibody. We can make this antibody now in the lab. It has that same kind of selectivity, and it's very selective between protofibrils and fibrils. And we think that that selectivity is really important for the therapeutics, potential therapeutics. Now we're studying inflammatory pathways. I won't go into these. I only have this one slide. It's just a whole nother area of research that we're trying to understand how this A beta is producing inflammation in the brain. So I'll kind of wrap up by showing that A beta and tau aggregation is very important. It can produce lots of different species. Studying that aggregation process and understanding the inflammatory pathways is really what's going to get us to where the therapeutics are that we need. Okay. It's been slow, and we all know that, and researchers are frustrated, but it's steadily making progress. And everyone works together and shares data at meetings and publications to kind of help with that progress. Okay. So there's lots of things that help support these studies. H has been very helpful in supporting a lot of our studies. The universities kick in a lot of funds to help, and there's lots of people that are involved in all of these studies at all levels of their educational process. So I'll stop there, and I will be happy to answer a lot of questions, I hope. And I'll stop sharing, but I can share that again at any time.
Thank you so much, Dr. Nichols. What really thorough and interesting presentation. One moment, please. Thank you so much. So we did get a lot of really great questions, and thank you everyone who joined us today. But first, I'd like to ask a few questions and maybe have a little bit of a brief discussion, and then we'll get to some of the questions from, maybe we'll even get new questions from the participants, the webinar registrants. First of all, so you were talking about the antibody that your lab has developed and the work that it is doing, or the way that it's you kind of attacking these proteins. What is the next step for that? Is what happens between now and potentially going to clinical trials?
Oh God, yes, great question. And I think probably everyone understands how long the process is for that to happen. But things like, let's say, this antibody, great in the research lab. We make that available to other researchers. A lot of times we just give them antibody, but we can also sell it on different vendors can sell that antibody. Pushing toward diagnostics and therapeutics becomes more expensive. And so there's always lots of safety tests that have to be done. More studies have to be validated in small animals. There has to be investors that are involved. So that takes people that are willing to take a risk and they think that your potential therapeutic has a possibility of working. So that's why it's so long in trials, because the safety is the most important thing. And after the safety becomes, does it work and how effective is it in, either preventing or reversing the disease? So this process can take 7 to 10 years. So some of the antibodies that were recently approved, I was reading papers on those antibodies 10 or 12 years ago. And so that process took that long. And it took companies to jump in and invest in that. So unfortunately, I mean, everything takes time and takes money for that to happen.
Yeah. Well, thank you. Can you walk us through a little bit about what happens in our body as we age that creates more of this amyloid beta protein, and what triggers that change?
Yeah, well, I mean, there's two kind of hypotheses, and both could be correct. As we start to slow down, not just our daily movements, but enzymes that we have in our body, and we have enzymes that make things, we have enzymes that get rid of things. So we clearly make this A beta every day, and we have enzymes that clear and get rid of it. The hypothesis is that as you age, the clearance enzymes start to slow down. Anything that you do that builds up A beta, the concentrations of it in the brain will promote its aggregation. We can see that in the lab. If we just increase the concentration, it aggregates more quickly. But also there's the enzymes that produce it. So if something impacts those enzymes and it causes them to produce more A beta in general or more beta 42, then that can also cause this aggregation effect. So I want to know that there are lots of people studying all of those aspects and trying to determine, does this happen in aging? Is there some other factor, environmental or nutritional, that impacts one of those two processes? And there's the aggregation, including us, to stop the aggregation, molecules that might be administered that can do that.
So I've heard
A
a
lot.
lot about the research around the tau protein. And I'm wondering what, besides your research, and I'm sure there's lots, what other kind of research is happening around amyloid beta protein and around those enzymes that kind of cut it free from the APP?
Yeah. So that has been going on for a while. And I think some of the frustration in the field is attacking the amyloid beta hasn't worked very well. My thought on that is not attacking, but companies jumped in a long time ago and tried to inhibit those enzymes, and they weren't able to be that effective doing it. So one of the theories is that by the time you see anything, it's almost too late. By the time someone has symptoms of Alzheimer's disease, for that time into play. And I think we would all love to be able to know what's going on, even to try to preempt things from happening. But I will say a lot of the research is focusing on protein right now. It kind of bounces back because people problem figure out the best way to stop it. And that tau is important.
So sorry. We're having a little bit of an internet lag here on my end, so I don't know if anybody else is, but I apologize for speaking over you. That actually is a really great segue into some of the questions that have been asked. Someone asked earlier, would you recommend to primary care physicians to add an APS2 blood test to a standard blood panel? Or not necessarily you, I know you're not a medical doctor, but is this something that we should do? Is this something that we should be screening for after a certain age? And are there other indicators that need to be met before we could request some sort of test?
Yeah, so it's not there yet. So it's not out. This is all data that was presented at meetings. People are writing it up for publications. So I'm giving like the most recent data that all the researchers are following. But absolutely, I mean, once that kind of is available and everyone and wants it to be effective. I mean, we are all competitive, but nobody's going to like stop. Everyone's working together. And what the rest of us would do would just be to try to make it better. It looks pretty promising. And as it's available, I would say the answer to this, and I would ask your primary care physician to try to, stay in tune to what's going on and, what is available at the latest things diagnostics wise.
Larry Spears asks, are there other protein aggregation diseases? And if so, are they limited to I'm going to mess this word up, neuronal diseases or other cell types that you're aware of?
A special topic class that we're on right now that studies all of these protein diseases. So ALS, multiple systems atrophy, degenerative protein diseases, because the process is very similar. There's lots of peripheral diseases as well that involve protein aggregation. So light chain amyloidosis, islet amyloid polypeptide, these are all in the peripheral system. And they create havoc and things like renal failure and cause dialysis. So protein aggregation in the peripheral system absolutely is a problem. And we study those. The process similar. It just peripheral tissues instead of neuronal tissues. Insulin also is a protein that aggregates and causes problems.
It's interesting you mentioned that. Somebody actually asked about, let me find the question, if there was a connection between, where is it? A connection between blood glucose and dementia or Alzheimer's disease. And whoever asked that, I apologize if I butchered it, but I think that was the gist.
Oh, no. And a lot of these things kind of go in cycles. So I would say about 10 years ago, everyone was looking at glucose levels, insulin levels, trying to find a relationship with insulin receptors, glucose transport. And every little aspect trying to link, we're always trying to link some sort of physiological system that might impact Alzheimer's disease. And it's kind of died away a little bit. I have a feeling it's kind of cycling up again. And all of this has to do with funding. So the more funding and the higher impact grant applications that are looking at a certain thing that they've maybe got some great data, preliminary data, that always research on that. We know that cardiovascular risk factors are correlated with Alzheimer's disease. We don't know exactly how the mechanism. No, if you have those risk factors, also seem to be risk factors for Alzheimer's disease. So same with diabetes, there's a correlation there. It's not as strong as people on say, but certainly we're paying attention to every potential thing. And we're always looking for high impact data that would suggest that.
Thank you. We have another question from Larry Spears. How would you time the administration of protofibril antibodies? And would the damage already done be done by the time someone with symptoms was treated?
Yeah, I mean, that is the catch, because nobody, so the administration of the antibodies is, monthly. It's an IV administration. Every medicine has some side effects, including there's some side effects to some patients with the antibodies. Nobody wants to have that done if they don't know if they're going to have Alzheimer's disease or not. And so that's where the diagnostics comes in. And yeah, we kind of are all working on parallel things, hoping the diagnostics get better, trying to develop the therapeutics. But certainly just wholesale administering expensive antibodies to everyone is probably not the strategy that any one of us wants to do. So yeah, you run into the problem. Is the damage too far done? So we're trying to look at, and it's in mice, and that's always a caveat, trying to look at ways that you can reverse the damage and not only prevent it. So there's always those two paradigms that you want to look at when you're doing the basic research.
Thank you. Barbara Green asks, is anyone conducting clinical trials of immune depleting therapies or anti-TNF type therapies in patients who are at risk for Alzheimer's to try and treat during the preclinical phase?
Yeah, targeting TNF has been around for a long time. And in fact, the rheumatoid arthritis, giving anti-TNF antibodies is one of the treatments. The thing is that I've focused on one inflammatory molecule. And what you really have in inflammation is lots of inflammatory molecules that are being produced. So I made it a little bit more straightforward, and I hope nobody was misled there. But TNF is one of many, many molecules that are produced. And so it's even the same with sepsis. Bacterial infection can cause the production of a lot of these molecules, including TNF. Those molecules in turn can also activate and recruit more cells. So it's really a cascade of events that happens. And so it's hard to target one of those. It's better to target the source that's causing the inflammation, because you might knock down one, but there's lots of others that are being produced.
That's why they recommend things like diet and exercise as preventative measures, correct?
Yeah, and anti-inflammation in general is always a good strategy. So it's it's in kind of that list of things that you want to do that hopefully will prevent Alzheimer's disease and, lowering inflammatory molecules, keeping active, keeping your mind active. Those are all on that list.
I think we have just one more question, and it is, if someone has lymph nodes removed in a surgery, could that cause inflammation that would trigger Alzheimer's? That seems pretty specific, but do you have any
That's
information on
quite specific, and I'm not sure
that?
if I can answer that question. Hmm. So it kind of asked about the microglia, and that was pretty technical because there are a lot of studies now, and I don't want to get too in the weeds here. There are ways to deplete immune cells, so microglia specifically. And these are, of course, done in small animals, not in humans, right? But it could be a strategy in the future. So if you knock out all the microglia, of course, those animals are going to be highly susceptible to infection, but they actually get a little better. And so a lot of the questions we have is, are the microglia actually contributing to the inflammation and the disease or in some other way? I mean, there's lots of people studying if they're impacting A beta directly. But that would be, so there's a lot of debate on what the microglia are doing. Are they bad? Are they good? It's probably both, but if it goes on for a long time, chronic inflammation is not good. So if you had a suppressed immune system, there would be lots of other issues that would come into play. And so that could be a risky type of manipulation there.
And I have one more that's kind of very specific. We had a user ask about cannabis use, and if you have any information about that contributing to Alzheimer's disease or to the amyloid beta protein buildup.
So I have not, I mean, I scour the literature every week. I get a prompt for anything related, any papers related to our area of research. And of course, that one would be intriguing, and I would scan that. I haven't seen a lot, though, related to that. People drilling down even further to look at the primary compounds in cannabis. So I don't know if I can answer that question
Okay.
thoroughly enough for you guys.
Well, thank you for all of the questions that you did answer. You have been just a phenomenal speaker, and I feel so much more, even though we had already talked, I feel even more well-informed about your work and about the processes that go into Alzheimer's disease. So I am very grateful to you, Dr. Nichols. Thank you so much for being here today. I also want to thank Stacy Portney, who is our Missouri Cures admin, who is here helping out in the background a little bit for us today. I greatly appreciate that. And then all of the attendees, I'm very grateful to all of you for joining us today. Wanted to remind you that our Lunch with the Researcher series, it is a monthly series. The next one is June 5th. We'll be featuring Dr. Paul Durham, who is the Provost and Fellow of Research at Missouri State University. And he studies migraine and pain, and he's a very fascinating speaker. So I think you'll really enjoy it if you join us next month. Dr. Nichols, is there anything you'd like to say before we sign off?
No, thanks to you and Missouri Cures for making this opportunity available, and thanks to everyone who joined in on the webinar today.
All right. Thank you, everyone. Have a great afternoon.